Okay, and I will turn it over to Steve, who will...
So the cryptic announcement was that this week, after several years of working hard on this venture, we announced officially the formation of the Berkeley Institute for Parks, People, and Biodiversity. John Jarvis is the director of that. You might recognize his name as the last director when we actually had directors of the National Park Service, when presidents would actually name directors of the National Park Service. John was a long-standing director for more than a decade of the National Park Service, and so we're hoping that this will be a great place to connect the kinds of biodiversity research that people do in places like this and elsewhere on campus with the work that we do in with people on campus and the work that goes on in parks and protected areas and other public lands. So we're very excited. Lots of opportunities we hope for undergraduates and graduate student funding. So look for more information on that in the future. It's a pleasure today to introduce to you Dr. Eric Riddell, who came to join my lab this summer. Eric did his undergraduate work at the University of Sewanee. And
oh, undergrad, you're right, sorry.
Undergrad. You've already blanked that part of your life out. Can you remember anything from the fraternity parties?
A little bit, practice.
Yeah, it was Tennessee. So. Yeah. And then did his PhD at Clemson University in Mike Sears' lab. Eric brings to us a wealth of expertise in thinking about physiological constraints on species ranges. And the reason why we wanted to get him involved in the Grinnell resurvey was we're working in a pretty difficult and harsh environment in the California deserts now. And Eric is going to be working on helping us to adapt the kinds of models he'll talk about today with ectotherms to thinking about the same problems with endotherms and their physiological limitations. So it's a great opportunity to learn a little bit more about what Eric did for his PhD, and welcome to MVZ and to Berkeley.
Thanks, Steve. And thank you for coming. I'm really excited to give this talk. It's kind of fun seeing all my new friends here too. So there are really three topics that I'm going to cover today and things that I really think a lot about, and that are what are the functional traits of an organism that support their geographic range? What capacity do those individuals have to actually adjust those traits, so phenotypic plasticity of these important functional traits? And what might that plasticity mean under climate change? My story actually begins 8 years ago when I decided that I didn't want to be a dentist, and I spent all of my savings and I moved down to the Galapagos to do some volunteer work. And I eventually got connected with this project on Nazca boobies with Dave Anderson out of Wake Forest. For the past 30 or 40 years they've been studying these birds in the Galapagos, and they've essentially been their population levels are fine, but their more famous cousin, the blue-footed boobies, they've essentially gone locally extinct on the island that we were working on. And the reason that they have at least found support for is that this Humboldt Current has been swinging wider of the Galapagos more frequently, taking these small sardines that the blue-footed boobies really need to actually feed on. Nazca boobies can eat the bigger flying fish and mackerel, so they've been okay, but boobies, blue-footed boobies, not so much. And I just remember thinking, like, that's so incredible that you could actually find this mechanism that sort of explained what happened to this local population. And that really started to get me thinking about what we know about climate change and an organism's relationship to climate. Now, historically, there are really two ways that we try to answer these questions. One is a statistical approach, or a correlative approach, that takes presence data of your species of interest, and you draw statistical associations with whatever sort of climatic or topographic features that you think are of interest to your animal to develop a statistical geographic range of your species. These models are really good at implicitly capturing all of the processes that might be driving your species: physiology, ecological interactions, and so these are typically more thought to represent the realized niche. But again, you don't really know what that's capturing, right? It's all implicit. As an alternative, people have turned to mechanistic, or more specifically, physiologically structured species distribution models to really understand what those functional traits are that support an organism's geographic range. So for these, you take how certain functional traits that are influenced by body temperature, for an ectotherm in this case, and combine that with climatic data in a sort of energy budget analysis, with the idea being that an animal essentially needs to balance their energy budget. They need to balance the amount of heat coming in and out, balance the amount of water that they lose, otherwise they die. And ultimately you can sort of project this across the geographic range that you're interested in to get an idea of where this animal could possibly survive, grow, and reproduce. Where can it actually live and get the energy for those processes? So the mechanistic models, this type, are typically more associated with the fundamental niche. Where could these animals actually persist? They typically over-predict right where the animal could live because it's the fundamental niche. It doesn't take into account things like competition. And why is this important? It's important because extinction rates are predicted to accelerate with warming temperatures. This is a review from Mark Urban in Science last year, showing how with each degree of warming, we're predicted to see this sort of non-linear response and extinction risk. However, most of the studies that were used to generate that relationship contain no mechanism. They're all statistical associations. So basically all that's telling you is that those statistical relationships between the presence data and the climatic data are basically falling apart. There were, several types of mechanisms that were investigated in this study, and that typically covered these 6 mechanisms that we often think about as ecologists. That includes species interactions, dispersal, which we'll talk a little bit about today, demography, evolution, the environment, and physiology. I spent most of my time thinking about how animals balance energy and mass budgets using their physiological traits, specifically with these small lungless salamanders that are fully terrestrial, and so they don't go to water to reproduce. They go underground, they lay little eggs, and out of these eggs hatch tiny little fully developed salamanders that are objectively very cute. And one of the things that I was thinking about is these animals are constantly bombarded by different sources of heat from their environment, whether radiation from the atmosphere, vegetation, the ground. They're also gaining and losing water with their environment. And I wanted to know, how do these animals actually balance this biophysical demand with their physiological traits to sort of be flexible and respond to their environmental conditions? So what's the sort of underlying theory about how these mechanistic models actually work? So we take climate and physiology, and we do this to predict the amount of time that the animal could be active. So this is universal across plants and animals. For instance, in plants, we might predict the climatic conditions where stomata can actually open and take in CO2 and release water and oxygen, or for something like a lizard, when it can actually leave its microhabitat and actually go forage and find mates, with the idea that that amount of time corresponds to how much energy that these animals can bring in, how much carbon that a plant can fix, how many seeds a mice can collect in a certain amount of time, and ultimately that might lead to some sort of estimate of their ability to survive and reproduce, some estimate of fitness. So I took this approach and I applied it to a very cool system just north of Clemson. So Clemson is right around here, and we were located right underneath the salamander diversity capital of the world. So there are more species there than anywhere else on the planet. And this is in the southern Appalachian Mountains. They are nothing like the mountains here. They're around 1,600 meters at my field site, so they're much lower in elevation. And what I have here is just a heat map showing you where the greatest species diversity is. And the black outline is this geographic range of the Plethodon jordanii species complex. There are 7 species within this complex. I primarily focused on the most widely distributed species. But all of these species can reproduce together, produce viable offspring, and they're all isolated on the tops of mountains. And that is thought to reflect their highly conserved climatic niche due to strict physiological requirements. So they like it cool and they like it wet. And they're all sort of trapped on the top of these mountains right now. And they can't tolerate those conditions between the mountaintops. So They're, like I said, I think I said they're nocturnal, they're fully terrestrial, they're lungless, so they do all the gas exchange between their skin and the, so that includes water and oxygen and CO2 exchange all across the same respiratory surface, their skin. That's the major parts of their physiology. In terms of their natural history, this modeling approach is very appropriate. So at night, they poke their heads out of their little burrows and they sort of determine whether or not those conditions are suitable for activity. If it's too cool or too dry or too hot, then they'll go back underground. It's actually pretty crazy. They all emerge at the same time, and if conditions are poor, they just go back underground, and they're gone for the whole night. If conditions are suitable, they'll be active. You'll find them just wandering around the forest floor, looking for food, like this cricket, or when the time is right, to find a mate. Now, one of the most important traits, or at least it's thought to be one of the most important traits for amphibians, is the rate at which they lose water, their water loss rate. And that's important for two reasons. The first is that it actually determines their body temperature. So it's the same reason why we sweat and we get a little evaporative cooling. These animals are very leaky animals, and they're always losing water. And so you actually get a little bit of an evaporative cooling effect as they're sort of walking around on the forest floor. And this is just an equation that helps you account for that effect of evaporative cooling. The other thing that, the reason that water loss is important is because it determines this time to dehydration. So these animals have, we've empirically found this threshold of how much mass they lose due to water loss before they cease activity and go back underground to rehydrate. So the rate at which you're losing water actually can constrain how long you have on the surface to forage. So the faster you're losing water, the less amount of time that you would have to be active on your forest floor. And if you could actually reduce water loss rates, you might be able to extend that amount of time that you have to be active in the environment. Now, when we measure water loss rates, this is typically what we use. It's called resistance to water loss. It's the same thing as circuit theory that we sort of learned in physics. We have resistance is equal to the gradient driving the flux. This is just property of the environment, how dry the air is. And then the cutaneous water loss rate. And this is what you actually measure in the lab. So how do we actually measure this? Here's my staged, I wear my lab coat every day. But so here's our little Plethodon metcalfi. We always take their mass before and after measurements. We put them in these flow-through systems that tightly control temperatures and humidities. We put them in this hardwire mesh to actually ensure that they don't behaviorally curl up onto themselves and limit water loss that way. We're trying to just get the pure physiology. And they like it dark, don't worry. And then we use all of these instruments to measure their water loss rate and volume of oxygen consumption in real time. So that's the biology, right? The other part of this equation is this part of the environment, and I have to do my little physics plug so that we all are on the same page. What really drives that gradient is something called the vapor pressure deficit. Typically plant ecologists are much better at this than animal ecologists. No offense, I'm an animal ecologist. So what that is we have temperature here on the x-axis. We have saturation vapor pressure on the y. And that's the amount of vapor, basically, at which the air becomes saturated at a given temperature. So you can see that it's exponential. And let's just pick an arbitrary temperature right here. And let's add in an arbitrary amount of actual vapor. What's left over is the vapor pressure deficit. And that's roughly about a 50% relative humidity. If the temperature were to cool, 80% relative humidity, and then 100% relative humidity. This is the dew point. This was kind of cool. It's kind of opened my mind about like why clouds form and there's condensation on your glass and on the windshield. But here's why it's really important experimentally. Both of these conditions are both 50% relative humidity. But under this warm treatment here, you would expect about a 2.5 times higher evaporative water loss rate from free water than in this condition. So the VPD is almost 2.5 times greater there. So that's why it's really important to control experimentally and think about the type of humid or hydric environment that these animals are experiencing in nature. All right, and then finally, for all the biologists in the room, there's really two components here. As a biologist, what I really want to know is what's going on in the skin. So this is just a cross section of salamander skin, and I want to know how these animals can actually biologically control water loss rates. But that resistance value is the sum of what's going on in the skin resistance, as well as the boundary layer. And so the boundary layer is, all of you right now are basically sitting in your own little microclimate, and you're exchanging heat and water vapor with your environment, and that flux, those changes, actually provide a resistance to heat and mass transfer. So depending on how hot you are or how quickly you're losing water, that can actually influence your rate of water loss. So we use a couple different methods, so basically what you want to do is get rid of this boundary layer, at least solve for it, so that you can get to the biology. There's two ways of doing that. The first is to use physics, which you basically use a lot of theory to figure out what the boundary layer should be given all of these different conditions. So the first value is the Grashof. No, I'm just kidding. I wouldn't do that to you. That would be awful. So you can use it doing first principles. You can also empirically measure it. So the idea here is you cast a model of your animal in agar, and then you measure this agar model the same way you would your animal, under the assumption that agar does not have a skin resistance to water loss. So whatever you measure is strictly due to the boundary layer. And then you can take, however, correct for mass, and however big your boundary layer here is for this size animal, you can actually solve for the skin resistance of your animal. So one of my first questions in graduate school is, well, how well do these actual methods agree with each other? And what might be the consequences of using either one? So I took all of these calculations that you can use, this theoretical approach, using dimensionless analysis, and I assumed the size of the cylinder, which is roughly pretty accurate under various types of convective conditions. And then I also made hundreds of these agar models, cast them in alginate, 3% agar replicas, which is what everyone uses. I use the same flow-through system that you've already seen, 5 different body sizes, 5 flow rates, I think 3 VPDs and 2 temperatures to see what we got. All right, so this is important. The next slide that you're going to see is I took both of these methods and then I applied it to actual salamander data to see what that meant for skin resistance, the biology. All right, so we have surface area on the X, resistance to water loss on the Y. Surface area is basically just, it's estimated from mass, so it's basically body size. And then we have our empirical approach in black, and then the dotted line is the theoretical approach. So the empirical is agar, and this is those physical equations. One thing you'll notice is there's a lot of scatter. That's largely due to the fact that these animals were measured at different humidities and temperatures. There's also geographic variation that's bundled up in here as well. But there was a couple troubling things for us. The empirical, the agar model approach, predicted that almost 48% of those values were negative skin resistance values, which by the definition means that they are losing water faster than free water, which from the perspective of physics doesn't make any sense. The other problem is that how the boundary layer behaved as well also really contradicted what we know about physics and violated the laws of physics as well. With the equations, we didn't get any negative values, and it also supported some of the biological patterns that we had seen in salamanders, suggesting that larger individuals have a higher skin resistance to water loss. So, and that's largely due to their, basically the vascular structure in their skin. So, people typically use this agar model approach. And under this approach, people have assumed, well, they have no skin resistance to water loss, which seemed a little unrealistic to us. So we were thinking, what's one way that we could demonstrate that, from an ecological perspective, that not having a skin resistance to water loss is potentially unrealistic, as opposed to having this little bit of skin resistance that we measured through the theoretical approach. So we built this first mechanistic species distribution model that incorporated various traits of the salamanders, so the rate at which they lose water, as well as their dehydration threshold, at what point they go back underground, their energetic cost estimated from metabolic rates, as well as their energy intake, which was a function of temperature and their energy assimilation rates. At various temperatures, so we use the microclim data set, which gives us a good idea of what a salamander experiences about a centimeter off the ground, combined with estimates of VPDs and soil temperatures when they're underground. We combined all these things to estimate activity budgets and energy balance over the whole summer over which they'd be active. assuming they didn't have a skin resistance and assuming that they actually had a skin resistance. So what did we find? So you're looking at the distribution just of the Plethodon metcalfi, the focal species, and you see here in this gray outline here the species range. All of these dots represent historic captures. There's somewhere around 6,000 captures. Dave Wake, who's not here today, he did a lot of these, which is kind of cool. And so what you're looking at is activity and energy balance without a skin resistance to water loss. And you can see that activity is really low. For an average night over that summer, they could only be active for about an hour, hour and a half, which just from being out in the field I knew was not realistic. Also, in terms of net energy balance, every single location was predicted to be a negative energy balance, which is also not sustainable. You have no energy left for growth, reproduction, or anything. So what happens when we add in a potentially more realistic skin resistance to water loss of just 5, so we go from 0 to 5. You see that activity time jumps up to 7 at some of the highest densities of where these salamanders are captured. And we also see that we start to get positive energy balance now, and again, these similar areas where these salamanders are most densely populated. So this was kind of exciting because it suggested that water loss rates could be this functional trait that sort of supports the geographic range of these species. And we had some physiological, biophysical, and ecological evidence that this might be the case. So this really got me thinking about what this means for climate change, because if these individuals can actually adjust these physiological traits in order to compensate or buffer themselves from the effects of warming and drying environment, then we might get a different idea of what happens under climate change. So what do we know so far? At this point, when I had started my degree, there was a paper by Milanovic et al. Who used that same Maxent statistical approach to predict what would happen under climate warming. He presented sort of they presented a best and worst case scenario of what would happen. Red is highly suitable, blue is not so suitable, and then they took this through time. And basically what you see is that even under the best case scenario, most places are unsuitable, and in the worst case, it's essentially all suitable habitat is completely gone. One of my main questions was to ask, well, how might plasticity and water loss rates actually change some of these predictions? And in order to answer that question, I had to figure out how plastic these traits were. So this was one of my first acclimation studies in the lab. So what I did is I went out to the field, I collected around 160 individuals in a stratified random design along a mountain. I brought them back to the lab. I kept them in their own individual containers where I kept them at a realistic cycling cool temperature for a month, just to acclimate them to their new conditions. After a month, I divided them up into a cool and a warm treatment. So the cool treatment was just the same cycling temperature, and the warm treatment was a little bit warmer to reflect some of the temperatures that they might experience under climate warming scenarios. And then at night, we did these simulated, and within each temperature treatment, there was also a humidity treatment as well. So 2 VPD treatments. So we actually cycled relative humidities with temperature in order to maintain the same VPD throughout the whole experiment. And so there are 4 treatments total. At night, we moved these individuals from their rehydration chambers, supposed to simulate them being underground, and we moved them to an open air chamber, which was actually soil, and it had an sort of a mesh lid to actually circulate the air so that they would actually experience that VPD. And we did this for 3 hours for a month. Every week we would monitor the animal's physiological traits with that same flow-through system that I would show you, that I showed you earlier, and to see what sort of responses we got to these acclimation experiments. So the first thing that I want to show you is that there was a lot of variation. So this is just a frequency distribution of how skin resistance changed over that 1 month period. We had about 10% that didn't change at all. They didn't do anything. They didn't budge. Most changed a little bit, and then we had some extreme cases where some individuals increased skin resistance or got more watertight by as much as 68%. So a pretty wide breadth of what was actually going on in this population. There's also no geographic variation. So it doesn't matter where I pick you up from. If I pick you up from the bottom of the mountain or the top, you have the same amount of plastic capacity. Now the best predictor of how much you change skin resistance to water loss was where you started out. So on the x-axis, we have your initial skin resistance, where you started off after that acclimation period. On the y-axis, we have the change in skin resistance to water loss. If you know, this is pretty crazy. We kept them in a saturated environment for a month, and these individuals didn't budge. They just stayed watertight at 7 seconds per centimeter. They didn't do anything. Other individuals showed this variation where if you essentially started off lower, at a lower skin resistance, you were more leaky. By the end of the experiment, you had increased your skin resistance to water loss by, again, by as much as 68%. So we had this sort of interesting almost variation or potential strategies for dealing with water loss. You know, do you sort of hedge your bets and always be watertight, or do you show some plasticity? So it also suggests there might be some sort of cost to being plastic, or that it's a little more dynamic than we think. So, oh yeah, okay, this is my little detour on mechanism. So I got a little bit of money to do a differential expression analysis on this experiment to figure out what could possibly be going on. So we collected individuals across this distribution and compared these individuals that didn't acclimate, so they maintained that high skin resistance to water loss, and individuals that were plastic. We did a very tissue-specific RNA extraction, so we just took the surface of the skin. We also took the hearts as well, but I haven't gotten there yet. And we took the skin because that's where we believed that, so this is where the action was going on. And then, you know, as far as I've gone right now, we've been just done DEC, differential expression analysis, and some GO-term enrichment. Most of the genes that involved, and by the way, we only found 54 genes that were differentially expressed between individuals that acclimated and didn't acclimate. That was pretty surprising, and I think that's because we gave them a lot of time to acclimate to lab conditions. We also spent a lot of work on controlling for humidities and things like that, and it was also a very tissue-specific analysis, so potentially that explains why we got so few genes. Most of the genes were involved with shutting off transcription, just thermal acclimation, shut down, and stop transcribing. There was this one gene, though, that was pretty interesting. We actually had it popped up 4 times of these endothelin receptors. So endothelin is a small peptide that binds to these receptors, and eventually through this cascade causes vasoconstriction. So this could explain these animals that were acclimating were turning this gene on relative to the other individuals that didn't acclimate, suggesting that this could be a potential mechanism. We know that perfusion has been suggested as one reason how how salamanders are actually limiting water loss, and so that's limiting blood flow to your skin. So this could potentially be a target. It's kind of cool because this is a big target for drug companies for hypertension. And so this could be, there's a lot of receptor antagonists that we could use to sort of play around with these receptors at first and just test if this is an adequate target to control water loss rates. And you can do all sorts of cool ecological experiments once we can demonstrate that. All right, back on track. So we know that these animals can exhibit an extreme amount of plasticity in the lab. What are they doing in nature? So over the summer of 2015, I measured the surface level humidities and temperatures that these salamanders experience over their period of activity from the spring in May to the fall in September. And then I brought these salamanders in at these same time intervals to see what their water loss rates were doing in nature. If we look at VPDs, we see the highest VPDs, or the driest air, happening in the spring, and then it sort of declines into the summer and it gets really wet for the rest of the summer as the rain comes in. So what were the animals doing? We see something that makes sense. So their highest skin resistance to water loss, around 7. Again, this is sort of a ceiling that we've found in our results. They don't really go higher than this. Started off at 7 in the spring when it was driest, and then sort of precipitously declined as it got wetter. So both in the field and the lab, they demonstrate capacity to respond to their environment in ways that make sense that would buffer themselves from a drying, warming environment. All right. So one of the things that I wanted to incorporate was this physiological plasticity and water loss rates. And so this physiology that I've been talking about for a while now, but also their behavior. So I mentioned at the beginning of this talk that they show this avoidance behavior of poor climatic conditions. So what we wanted to do was integrate that sort of behavioral avoidance as well as a potential strategy to minimize the ecological impact of warming. All right, and this is just a schematic of roughly how the model works, this physiologically structured model. We start with those estimates of relevant surface temperatures. We run through every single pixel, and we correct for adiabatic lapse rates. Basically, the resolution on this is pretty poor, and so we just correct using adiabatic lapse rates, which is how much the air cools as you go up in elevation. We did this for every year from contemporary climates to the end of century, assuming business as usual warming, so we just continue as we don't do anything to stop or curb carbon emissions. The model assigns individual traits to the animal. So first is body size, and we tried to cover the range from juveniles to adults. It also assigns a skin resistance to water loss, where we took the upper and the lower bounds of that physiological plasticity that we had seen in the lab and in the field to say what's the most that physiological plasticity, at least in water loss rates, could contribute to buffering the impact of climate change. It also assigns a behavioral strategy, either where you maximize activity time or you try to maximize energy balance. And we did that by saying, if you're a salamander and the net energy balance of being active is lower or worse than just going underground, go underground and just stay underground. Even if conditions might be within your thermal range to be active, if the costs are too high, just go underground. So the model runs through an average night for every month over the next century, using a combination of these biophysical, the boundary layer dynamics, as well as the physiology that we measure in the lab, which also includes the thermal sensitivity of metabolic rates, as well as energy assimilation, and predicts activity and energy balance for every pixel throughout the whole range, as well as the extinction. And we did extinction, we modeled extinction in two ways. One was whether or not the animals actually experienced their thermal limits, and another was whether or not they depleted all of their lipid reserves. So we estimated lipid reserves by the average number of triglycerides that a salamander has for a given body size, and once you burn through those lipids, you die. And a spoiler alert, every single individual went extinct for that reason, for balancing energy budgets, and they essentially went extinct before ever experiencing anything close to their critical thermal limits. All right, so let's get to the model. So what you're looking at here is energy balance across the whole species range. We have the species range in black. Anywhere in green, they are predicted to have enough energy to reproduce, to support their biennial strategy for reproduction. Yellow is 0, and anything orange and red is a negative energy balance. Roughly about half of the sites in this scenario could support a salamander, and this is without acclimatization or avoidance behavior. So that plasticity that I was talking about is not incorporated. So if we don't incorporate plasticity, what do we see under climate warming? By 2050, we lose about half of their suitable habitat, and by the end of the century, it's almost all gone. Okay, let's go back in time. And let's start back under contemporary climates. Now let's add in acclimatization and that avoidance behavior that we think is important for structuring their habitat, their species range. And we see that under this best case scenario where they can maximize activity, 90% of the regions within the species range can support these salamanders, except for this really sad small little nub here, and that's actually where Clemson is located. So that was a little frustrating, but they're really rare down here, and they typically stick to seeps. So what this is kind of telling you is this model does a fairly poor job at predicting that really small microhabitat. But otherwise, it seems to do an okay job. As we continue through climate warming, what do we see? The range contracts by about 15% by 2050. And by 2100, we've seen about a 30% range contraction from the beginning of the century. But the large part of their core, even in the Great Smoky Mountains here, and especially here in the Balsam Mountain Range, those seem fairly suitable even under the worst case scenario. So we kind of have the worst case warming scenario with the best case biological response, and it looks okay, at least in the range. I mean, it's much better than losing everything, right? And I'd like to point out one thing. If we go back to contemporary climates, and just keep your eyes here in the core of the range, the model actually predicts that the core of the range will have more energy by the end of, due to climate warming, but sort of like the energetic costs eat away at the edge. So if you're in the core, a little bit of warming appears to be a good thing. It allows you to bring in more energy, but quickly energetic costs overtake that energy gain that you see, and you go extinct on the edges. And like I said at the beginning, we varied body size in this analysis. And it explained the 52% of the variation that we saw through all of our these iterations of this model. So we have time on the x, we have proportion extinct on the y, and this is for a 5-gram salamander, which is about as big as they get for this species. And then we've laid out no acclimatization, just physiological acclimatization, and then both physiological acclimatization and avoidance behavior. And if you're a big salamander, so 0 means everyone's fine. You're pretty much unaffected by climate warming. But as you get smaller, you see that you become more sensitive to the effects of climate warming. So as we get to a 2-gram salamander, by the end of the century, we see that under all these different scenarios, everybody kind of ends up at the same place. So what this suggests is clearly these large individuals would have to get through small life stages to get big. But what this is telling you is potentially that these juvenile life stages might be experiencing the most physiological stress under climate warming. So one of the questions that we wanted to ask was, what sort of physiological response would it take to get from here back down to contemporary levels of habitat suitability by the end of the century? And so we ran a sensitivity analysis to ask that question, and we kind of came up with 3 scenarios by which juveniles could essentially maintain that same level of habitat suitability. This first one is adaptation of skin resistance. So this is the frequency distribution of skin resistances that we saw from our experiments. And in order to maintain habitat suitability, these juveniles would have to have a value of 9, which is much higher than what we see. In terms of energy assimilation, we essentially try to maximize that function by just taking one standard deviation of the relationship that we saw. So this is average and this is maximum. That restored energy balance. And for energetic costs, we asked whether or not individuals could actually reduce metabolic rates, or the sensitivity, thermal sensitivity of metabolic rates as one way, and a reduction of about 20% got you there to restoring habitat suitability. All of these scenarios require some pretty big assumptions, though, to get there. We have no idea that this is even a heritable trait or there's any genetic variation just for selection to act upon. Maximizing energy intake, that's kind of unrealistic as well. And we just, who's seen the new 80% decline in German insects? So this will depend way more on prey abundance, and we assume that prey is just readily available in our model. As long as they're active, they can be eating the whole time, because otherwise it's a very hard thing to model. And the last thing is energetic costs. In order for this to work, these salamanders would have to reduce metabolic expenditure throughout the whole year. And it's unlikely that they'll actually get that thermal cue to reduce sensitivity throughout in these cold months. So they need to reduce metabolic rates in cold months, which is probably unrealistic. Okay, so this problem with the juveniles, this is for my last part of the talk, really started to get me think about a broader temporal perspective. We know that salamanders have lived through some pretty intense climatic change even over the last 1,000,000 years. And so can we use this model to understand what they might have done in order to survive during these time periods? So what you're looking at here is variation in surface temperatures over the last 800,000 years. And you can see the Milankovitch cycles as the Earth sort of tilts and swings wide of the sun. And so we wanted to use the same physiological approach to understand how energetics and activity times potentially changed throughout the year during the last climatic oscillation. So this is the last Pleistocene warming, the last glacial maximum, and then back to the Holocene. So what do we see? So this is just that same time scale blown up. This is the peak of the last Pleistocene warming. And then here we're showing net energy balance and activity time. And you can see that, so month of year explained most of the variation in our data set. And basically during the warm months, you saw these heavy energetic costs. And then for activity time here in blue, you see this bimodal pattern of activity. As we go to the last glacial maximum, you see that energetic costs are much lower, lower energetic costs, higher energy gain, looks to be more suitable. And now you have this unimodal pattern of activity where July is the month to be active. That's the most suitable time to actually to be active. As we begin to warm again, we see that this bimodal pattern start to come back, and then under Anthropocene warming, we see it even kind of push it even further. So what's kind of cool is this physiology gives you an idea, or at least a prediction, of how phenology changes, strictly based on physiological traits, which is kind of cool. That was kind of exciting. Because we know that changes in phenology are an important part of responses to climate change, and can be predicted from physiology. So what was going on spatially? Same map as before, except this is in the last Pleistocene warming. We can see that net energy balance is highest at these high elevations, similar to what we have now. They're basically restricted to the tops of mountains. Across latitude, no real change. And if we look at the last glacial maximum, it looks kind of nice. There's a lot of green, right? So it's pretty very different environment where now the low elevations are positive energy balance, so they're suitable, and low latitudes are more suitable than high latitudes. And I just want to note that not a single area, geographic region, was predicted to run out of energy in this case. So it's a time of low energetic costs. What you'll also notice is where they lived before is no longer suitable. So as we know, kind of what happens during these climatic oscillations is animals have to disperse out of these areas to new suitable habitat. And salamanders are really poor dispersers. So the average distance is around 1 to 2 meters. There's a nice distribution. It can get as long as 10 meters. And so one of our questions was, well, how far could they have gone in this time period? And what's kind of interesting is when they were standing at the tops of these mountains in this warming period, down to the next glacial maximum, they had about 130,000 years. So even though they're only moving a meter or two, if you do this for 130,000 years, you're going to get pretty far. But then at the last glacial maximum, it warmed very rapidly. So you could have this long time to sort of march out, and then you had this rapid rise in temperatures where you had to get back to the tops of mountains. So how does that sort of dynamic work out? We built our Salamander diffusion model, which incorporated that distribution of dispersal distances, as well as the minimum age at reproduction, and their biennial strategy for reproduction. So they reproduce every other year. And so what do we see? So these areas in blue represent where the animals were at the last Pleistocene warming on the tops of mountains. And this area in green represents how far they could have dispersed over that 130,000 years. And it's about 40 or 50 kilometers or so. And then these different dots here are some of Dave Weisrock and Larson's haplotype data. It's a multi-locus mitochondrial haplotype. And essentially, we were just overlaying this data to demonstrate that this could be one way to actually predict population differentiation in terms of looking at these long-distance colonization events and continuous range expansions. So the analysis that we're trying to work on now is basically trying to predict FST values by incorporating physiology into estimates of resistance across the landscape. So this was during that last long period. What happens when it gets warm again? So now only the animals in blue were close enough to the tops of mountains to actually make it back up to the top. Everywhere else in red was actually predicted to go extinct. They were too far away to actually get to a place that would be suitable in the future. And so for my last sort of note at the end of this is, what does this mean potentially for conservation? The Nature Conservancy has developed this plan to conserve the stage, not the players, with the idea being that we conserve the regions of highest geological diversity under the idea that those support the highest biodiversity through these climatic oscillations. So I essentially wanted to test that idea. These areas in blue are the protected areas. And basically I wanted to say, how well do these areas in blue capture those dynamics of these animals moving up and down mountains through climatic oscillations? And this is what you find. Areas in blue are within the TNC's protected regions, and the areas in red are outside. And then we sort of, we added this priority index where we just bounded it by energetic gain. So areas that had the highest energy balance were assumed to be of the highest priority for conservation, and the lowest were the lowest. And then we broke these down by pie charts just to give you an idea of what was going on. So they missed about 63% of the total area that is supposed to basically harbor these dynamics for the salamander capital of the world. We're especially poor at low elevations at capturing that, which makes sense. That's where we live and grow our food. And we're a little better at higher elevations. And then across latitudes, it's split. So you can sort of make your own interpretations about what that might mean. I realize that the planet is warming, and but eventually we will experience these oscillations. So it was really just a test of their idea about how well geological diversity captures biodiversity through time. Anyway, so that brings me here. So now I'm in Steve's lab, and I'm really excited to work on the Grinnell Resurvey project. I'm particularly thinking about how endotherms deal with such a harsh physiological environment in the deserts, and building these models to really figure out what sort of heat loads that these animals have to deal with, and whether or not their physiological capacities can match those biophysical demands. So really excited to be here. Thank you all for for your attention. And I'd like to thank the following people. My committee members are fantastic, all my funding agencies, and thanks again for your time. All the failures that I had. Because who wants to hear about that? We had done this experiment twice before and had an idea of that's about how long it took for all the individuals to stabilize and stop doing anything. That data is not published and may never see the light of day, but I learned a lot about how to do the experiment correctly, and that seemed like an adequate time to in terms of gene expression, we had no idea. But in terms of physiology, that seemed like a reasonable amount of time. I worked with Kelly Zamudio on the gene expression data, and she also recommended, she said, no, about a month is pretty good. Yeah, sometimes I do 2 weeks, but since I already had data on what was going on, then I had a good idea of how long acclimation time should be. Yeah.
With your future climate models, did you take into effect rainfall differences that they're expected to happen?
Yeah,
Okay.
I didn't. So that's a really interesting dynamic because the southern Appalachians are predicted to get wetter.
Yeah.
And however, one of the interesting thing about global circulation models is that they make the prediction that relative humidities are predicted to be the same across climatic change, even these glacial periods. It can be a little bit somewhat contentious, but that's what these global circulation models say. So we assumed that was the case, that relative humidities stay the same. But with warming, as I showed you guys, if relative humidities stay the same and it gets warmer, it's actually still drier because you have a larger vapor pressure deficit, because that increases exponentially. So we operated under that assumption that relative humidities maintain the same. So.
But if it's raining more, then some of the areas they live might be flooded and they
Totally.
can't live in water.
Totally.
Right?
And so what's kind of cool is part of what we were doing was like the worst case climatic situation against the best case physiology, right? So if you're right, then we really have nothing to be worried about. But what we're trying to provide some confidence that they do have some plasticity in their behavior and physiology that could buffer them. And what's nice about these maps is they're very spatially and quantitatively explicit. So really what I see them as a very explicit hypotheses to continue to test. So now that we have this map, we can go out into the wilderness and actually see if individuals in the core of their range do bring in more energy and things like that. Yeah?
I was curious about if you've gone out in the field and looked to see if juveniles do something different from adults, given that you predict that juveniles should have a different response.
Yeah, so it's a great question. So juveniles typically, like I said, they hatch to the size of your thumb. Until they're about a half a gram or so, which is about a year of growth, they don't really come out on the surface. So we didn't get into that system because we don't really know what's going on there. They just stay underground, and I'm sure they're eating tiny little mites or something, and eventually they come out and are active on the surface after about a gram or two. So that's typically when you start to see them, and that's when we're suggesting you see the greatest amount of physiological stress. It could be you're right. Maybe these individuals just say, we're going to spend more time underground until we're bigger. Then the question is there enough food underground to actually support that growth? I don't know. I would imagine since they're coming up to the surface that there is some data from the 50s showing that individuals that are more active grow more rapidly and gain more mass. So activity on the surface is important to get that extra energy. So it's a good question.
So do you expect the size relationship that you've detected here to apply to all salamanders?
No, yeah.
You wouldn't? So you think that
No.
other salamanders would the juveniles
Well,
wouldn't be more susceptible? Is this not like a component
Oh,
of...
okay. This specific system was selected for a reason. And that was because I didn't have to think about aquatic larval stages. And a lot of these other salamanders that represent that diversity have that aquatic larval stage, and I think that's a totally different realm of selective pressures that they're going to have to deal with.
Yeah, 2/3 of all salamanders are plethodontids.
Sure.
Lungs.
Sure,
Some of them have larval
they're
stage and some of them
right,
don't.
right, exactly.
So a lot of them don't.
But I'm saying across the diversity of the plethodontids, you have all these different life history strategies, essentially. So I would never make a blanket term. But I would say that these species are often some of the most abundant species in the area. When you walk out there, they represent the majority of the biomass in a large part of the region. So that's where I kind of make that selling point, is that in terms of biomass, these are very important. Across species diversity, you'll probably see all sorts of responses based on their life history strategy.
So part of my reason for asking the question is I'm just curious if you would think that smaller salamander species are actually under more threat than larger species, or whether it doesn't matter because even the large ones have to go through the small stage.
Right.
At least for these direct developers that don't have an aquatic larval stage.
My gut is saying more the latter. That doesn't matter. You still have to grow through that stage, and it depends on whatever strategy that it takes to get you there. So, like if you're buffered under soil or in the creek from these sort of climatic variation, it's a totally different environment than being on the forest floor. So, yeah, I would say the latter.
Can I ask one more question?
Yeah, of course.
Same line.
Yeah, sure.
So is anybody doing a Barry Sonerbo type analysis of salamanders and other amphibians to see if we might actually already be seeing an effect where smaller species are lipping out?
Dave was on a paper in Central America showing that essentially detection rates have declined significantly with Sean Rovito.
I know that paper, but I think they're more concerned about chytrid in that case than they are physiology, right?
They
It's
do.
not a
Well,
size
in the paper they
relationship.
allude to climate change. I mean, if you look in the discussion, they're like, we don't have any other indication of something else going on, and maybe climate change is a silent killer. In the Appalachians, Nick Caruso's done some work where he's gone through and looked at detection and occupancy across the range, and has concluded that salamanders are becoming less, or these sites are less occupied under current climates, and that they're shrinking. Both of those papers are highly contagious, and they experienced a lot of backlash for reasons mainly to do with museum specimens. People were worried about non-random sampling across museums and interpreting incorrectly that you're getting this biological response when it could just be statistical. So I would say we don't know, and the ways that we've tried so far to deal with those long-term data sets have a lot of issues. And so I think what these maps allow us to do is potentially to go out there and test some of these ideas. So anyway.
The trick is apps.
Yeah. Exactly. Yeah.
So one thing I thought was interesting is the agar model
Yeah.
conundrum.
Yeah. Thank you.
Do you think that that's,
It's been a lot of time.
is that just that when you start to evaporate agar models, they like make their own skin on the model?
So are you asking like why are agar models
why
poor?
doesn't that work?
Yeah. So there's a few reasons. One, yes, agar clearly has like temporal limits, right? Eventually it dries out and it'll stop reflecting the conditions that you want it to. The other one is agar might actually have a skin resistance. That assumption that it doesn't have one is false. And that makes sense. Agar is really good at holding onto water. It has all these covalent bonds that it makes with water, and so that it's like, or with itself, and so it's holding onto water really well. The other is there's a problem with how we measure water loss rates, and that is that we assume that that water vapor density gradient is the same across all flow rates. But people measure these things at really low flow rates, and the chamber actually gets saturated. So you're assuming a value, this gradient, that's actually false, proceeding with these calculations. Nobody ever validates it. Nobody ever has. And you're proceeding with these false calculations, and you're more likely to overestimate the boundary layer of these animals, and thus conclude that your animal does not have a skin resistance to water loss.
So should you use larger chambers, or what's the...
You should. It took me 5 years to come to that conclusion. So, there's a few things you could do. You could either build a larger chamber, or you could increase flow rate size. And you could actually just experimentally look at where sort of that water loss rate plateaus, and then you've reached some sort of range of flow rates that seems appropriate. So, yeah, great. I love that question.
All right, on that note, let's thank Eric one more time.
Thank you.